Okay, so chapter 16, we're in November, already in chapter 16, crazy. So blood is your internal transport system. It's the life-sustaining transport vehicle of the cardiovascular system. That's because it flows all of your hormones, your oxygen, your carbon dioxide. Its function include transport of all of those molecules, the regulation, so in terms of blood pH regulation, blood pressure regulation, and also protection has to do with your leukocytes or your white blood cells, which are part of your immune response. So here are the transport functions. As you guys know, probably by now, it delivers oxygen and nutrients to all of your body's cells. And then in doing so, when it delivers its oxygen, it picks up carbon dioxide as the waste product of cellular metabolism and takes that to your lungs. When you exhale CO2, that's you're eliminating the waste product of cells. And your kidneys will also filter the blood daily to get rid of any harmful substances. It also transports hormones from all of your endocrine organs to their target tissue or organ structures. Regulation, so your blood functions to maintain body temperature by absorbing heat and also distributing it. It maintains normal pH using buffers, which are just alkaline reserves of bicarbonate ions. And it also maintains adequate fluid volume to help with blood pressure in the circulatory system. Here are the protection functions. It prevents blood loss. So you have platelets as a cellular component in your blood that helps to initiate clot formation. Blood also helps to prevent infection. Agents of immunity are carried in your blood. Your antibodies are in your plasma. You have complement proteins, and also your white blood cells are your leukocytes. And you have 5 different types of white blood cells. Blood is your only fluid tissue in the body, so we call it a type of connective tissue. The matrix is the non-living fluid, and we call that your plasma. Plasma is mostly made up of water, ions, and then cells are the living blood cells, and we call all of the cells in blood the formed elements. And your cells will be suspended in the plasma, and these are your formed elements. Your erythrocytes are your red blood cells. The leukocytes are your white blood cells, and the platelets make up that third part of those formed elements. So if we look at the composition of blood and we were to take a sample out of you and then spin it down really quickly in a centrifuge, we would get 3 different layers. The erythrocytes are the densest, so they would sink to the bottom, and they make up about 45% of whole blood. The hematocrit is a blood test that tests for the percent of blood volume that is red blood cells. So the hematocrit will always be a percentage. And here are the normal values of hematocrits. Females are slightly under males or smaller than males in their hematocrit values. And then the Buffy coat makes up less than 1% of a blood sample. And this is where your white blood cells will be found and all of your platelets. And then the plasma will float to the top because it's less dense than the erythrocytes. And that makes up about 55%. So here's just a picture showing the major components of whole blood. Again, spinning it down with the centrifuge allows the erythrocytes, the denser cells, to sink to the bottom. They're about 45% of a sample. And then this buffy coat is this thin kind of whitish layer. It makes up less than 1% of the sample. And this will be where all of your leukocytes, again, those are your white blood cells. They help with the immune response, and your platelets will be. And then the plasma floats to the top, and that's about 55%. Some physical characteristics: it's sticky, it's an opaque fluid, so you can't see through it, a metallic taste. The color will vary with oxygen content. So high oxygen levels will show a scarlet red, and low oxygen levels show a dark red. So that's kind of this idea of where we color arteries red and veins blue in drawings, because there is a difference in color between blood vessels that are high in oxygen or low in oxygen. And I shouldn't say that, because some veins are high in oxygen, and those are your pulmonary veins that come from your lungs. But that's the idea of where this color scheme comes in. Normal blood pH is right around 7.4, 7.35 to 7.45, I guess they say. Blood makes up about 88% of your body weight. And on average, we have about 5 liters, give or take 1 liter if you're a male or a female, which is a lot of blood or a lot of volume in your body. The blood plasma is the straw-colored sticky fluid. It's mostly all water, and then there's other dissolved solutes in plasma. Your hormones will be in there, nutrients, gases, wastes, proteins, inorganic ions. You'll have different proteins in the solutes of plasma, and albumin is the most abundant plasma protein. I just like to point that one out because you'll learn a lot of stuff in blood, but albumin is one of the most abundant plasma proteins. So here's just a good kind of summary table of the composition of your plasma and what it makes up. And again, most of it is water. You'll have electrolytes. Albumin, again, is the most abundant plasma protein. So that is plasma. So then we'll go to the formed elements, which are your RBCs, white blood cells, and platelets. Only the white blood cells are what we call complete cells. And that's because your red blood cells have no nucleus and they don't have any other organelles. So your red blood cells, their only purpose is to carry oxygen. So they have hemoglobin in them. Otherwise, your white blood cells are called the complete cells because they'll have a nucleus, lysosomes, other organelles in them. And platelets are just fragments of cells. Many formed elements survive in your bloodstream only for a few days, and most blood cells will originate in bone marrow and they will not divide. So this is a look at the formed elements in blood. Here are your leukocytes. The erythrocytes are the biconcave disc-shaped cells, so they look like donuts, but without the hole in the middle. Platelets are the smaller kind of cell fragments. So those are the blood cells. And this is a look at a blood smear. I'm sure you guys have seen this in your histology views of anatomy. Erythrocytes make up the majority of the histology blood smear, and then you'll have the white blood cells will all be the different stained ones, depending on what stain is used. And you can see some of these leukocytes have granules in them, which are tiny little specks, and others do not. Some of them have a bilobed nucleus or a multi-lobed nucleus, and we'll go over the different types of leukocytes and what they do. Okay, then we get to talk about erythrocytes. Are we working from Pearson yet? There's a PhysioX lab, Nakasha. Is that your question? There's a PhysioX lab coming up.
Yes, because I know I purchased it, but I'm not sure if you guys have been, or if the class has started working on Pearson just yet.
We've had, I think, one PhysioX lab before. I don't know if you completed that one. That was in the last module.
I think I did, yeah.
Oh, good. Okay. In terms of like other resources, there's a lot of other resources in Pearson, like the dynamic study modules. There's a lot of like questions that you can use to quiz yourself. I think a lot of people just work through a lot of the like quizzes and questions.
Okay, thank you.
Yeah, and if anyone else wants to say how you use Pearson, you can enter in the chat, but I hope it's been beneficial. That'll be my question for you at the end of the semester if it has been.
Okay, thank you.
Thanks for asking, yeah. All right, so erythrocytes and how they transport oxygen and carbon dioxide. So these have a small diameter, so they're extremely small cells. They have a biconcave disc shape. They are anucleate, no nucleus, no other organelles. All they have is this HB protein molecule, this hemoglobin protein that is designed for gas transport. Red blood cell diameters are larger than some capillaries. So they're able to kind of squeeze through some of those capillary blood vessels. They're a superb example of complementary of structure and function, and that's just describes how its structure is completely related to its function. So the biconcave shape gives it more surface area. Hemoglobin makes up 97% of the cell volume. Red blood cells have no mitochondria, so ATP production is anaerobic, so they don't need to consume the oxygen they transport, and that's really important. So your red blood cells can function anaerobically, so they don't need to consume any of the oxygen that they're transporting. So here's a look. You can see this biconcave. That means there's an indentation on both sides. That gives it more surface area. Hemoglobin will bind reversibly to oxygen, meaning it can unload oxygen and reload it back in. These are the normal values of hemoglobin. So hemoglobin is another test that they can test your blood sample for in terms of grams per milliliter, how many hemoglobins you have. Hemoglobins consists of a red heme pigment bound to the protein globin. This globin is composed of 4 polypeptide chains that look like that. The heme gives it its red color, and each heme central iron atom can bind 1 oxygen molecule. So here is a hemoglobin structure. It's made up of these kind of 4 chains, these 4 globin chains, 2 alpha and 2 beta. And then each heme group within each chain can bind 1 oxygen. So this just means that 1 hemoglobin molecule can bind 4 oxygens because there's 4 chains to it. Here's another look at the hemoglobin molecule with iron in the middle. So iron is an important element to hemoglobin. So we'll talk about people who are anemic, and sometimes low iron has to do with that because iron is important in the hemoglobin structure. So each hemoglobin molecule can transport 4 oxygens, and each red blood cell contains 250 million hemoglobin molecules. So we're talking about 1 red blood cell carrying 1 trillion oxygen molecules, if you're doing that math, which is just crazy. 1 tiny red blood cell can carry 1 trillion oxygens. Oxygen loading in the lungs produces oxyhemoglobin, which gives it a ruby red color. And oxygen unloading in tissues produces deoxyhemoglobin or reduced hemoglobin, and that's a darker red color. So again, these colors come into play, not only the color of the hemoglobin, but also the blood. Carbon dioxide loading in tissues. So 20% of carbon dioxide in blood will bind to hemoglobin. The other carbon dioxide is transported in the blood as bicarbonate ion that we'll talk about, but 20% of it is bound to hemoglobin as carbon dioxide. Hematopoiesis is just the formation of all blood cells. It will occur in your red bone marrow. Hematopoietic stem cells are stem cells that give rise to all the formed elements. So this just means that all of your cells or your blood cells, whether they're a white blood cell, a platelet, or a red blood cell, will all arise from one kind of precursor stem cell. Stages of erythropoiesis. Erythropoiesis is the process of formation of red blood cells. It takes about 15 days. There are stages of transformations. It starts with a hematopoietic stem cell and then will differentiate eventually into the red blood cell. I won't ask you guys to know the different stages of the production. It's interesting, so I included in here if you want to look at it, but eventually we get to a mature erythrocyte after we get through these stages. And this just shows you how here's the initial stem cell and how once it commits, so once it kind of forms past a certain step, that commitment step will go on and form an erythrocyte no matter what.
Earnings guarantee, healthcare, and accident.
All right, I'm just gonna hear more election TV. Okay. Regulation and requirements of erythropoiesis. Too few red blood cells lead to tissue hypoxia, and too many red blood cells will increase blood viscosity or the stickiness. More than 2 million red blood cells are made per second, and the balance between red blood cell production and destruction of red blood cells will depend on hormonal controls. So your hormones will kick into gear to create red blood cells, and also your dietary requirements will play in that. So the big hormone that I want you to know that's responsible for red blood cell production is erythropoietin, sometimes abbreviated that EPO. It's a hormone that stimulates the formation of red blood cells. There's always a small amount of this hormone in the blood to maintain the basal rate because your red blood cells need to be continually made. And erythropoietin is released by kidneys, some from your liver, in response to hypoxia. And hypoxia just means low oxygen levels. So at low oxygen levels, this erythropoietin will be released from your kidneys to stimulate red blood cell production. Causes of hypoxia will be decreased red blood cell numbers due to a hemorrhage, so bleeding either internally or externally, increased destruction of those red blood cells, insufficient hemoglobin per red blood cell, and this is where iron deficiency comes in because iron makes up hemoglobin. If we have insufficient hemoglobin, usually due to maybe an iron deficiency, that will cause this erythropoietin to kick into gear to try to create more red blood cells. Or at reduced availability of oxygen. So if you're at a high altitude, my family's moving to Quito, Ecuador, and we're going to be at almost 10,000 feet. So our body will probably have take several days to kick in to make more red blood cells. Or lung problems such as pneumonia. Too many erythrocytes or high oxygen levels in blood will just inhibit the erythropoietin production. Erythropoietin will cause erythrocytes to mature faster. Interesting to note, testosterone also enhances erythropoietin production, which gives males usually that higher red blood cell count, which will contribute to their higher hematocrit numbers as well. Okay, so this just goes through kind of this negative feedback balancing mechanism of homeostasis of normal blood oxygen levels. If oxygen levels decrease, as in hypoxia, that'll lead to a decreased red blood cell count, decreased amount of hemoglobin, and eventually the decreased availability of oxygen. Your kidneys then will release erythropoietin, which will stimulate red bone marrow to release more red blood cells that will increase the oxygen carrying ability in your blood, and that will rise to get your homeostasis balance back to normal. Some athletes abuse artificial erythropoietin, and I am not an expert in what athletes use in terms of drugs, so don't ask me any more questions about this. But from what the textbook says, some athletes will abuse artificial erythropoietin, and erythropoietin will just increase their hematocrit. So why would athletes want to pump themselves with erythropoietin? Why would they need that? Anyone want to write in a chat if you're not watching the results or answering?
Would that be because to get more oxygen into their blood and into the muscles so they can perform better?
Exactly, yeah. So more red blood cells just means more oxygen, and more oxygen will just increase the really how well their cells can create ATP. Oxygen is needed by all your cells to create energy. And if you can get more oxygen to your body, you create more ATP, and more AT means your muscles can contract more quickly, faster. Yes. There's dangerous consequences because it can increase your hematocrit too high, with dehydration concentrating blood even more. So blood could become like sludge, and that sludgy blood could cause clotting, stroke, or heart failure. Not worth the risk, probably. Dietary requirements for erythropoiesis. You need amino acids, lipids, carbohydrates, but iron is the big one that you need from your diet. 65% of iron is found in your hemoglobin. The rest will be either stored or being recycled in your liver, spleen, and bone marrow. Free iron ions are toxic, so iron will always be bound with proteins, stored in those proteins. Vitamin B12 and folic acid is also necessary for DNA synthesis for rapidly dividing cells like red blood cells. So iron, vitamin B12, folic acid, if you take any supplements, those are probably good ones. Fate and destruction of erythrocytes. So they don't last forever. Their lifespan is at most 120 days. They're anucleate. So because they don't have a nucleus, you guys know that your nucleus is where all new proteins are synthesized from DNA to RNA. So because red blood cells don't have a nucleus, they cannot grow, they can't divide. So they are destroyed. They will degenerate themselves. They can get trapped in smaller circulatory channels, especially in the spleen. Macrophages in the spleen will engulf and eventually break down the dying red blood cells. But your red blood cells, when they're broken down, their components within them can be recycled and stored, like their heme group, the iron and the globin, parts of the hemoglobin. Heme can be degraded to bilirubin. Livers will secrete the bilirubin as bile into your intestines, where it will be degraded even further down. and that will leave your body in the feces. And globin will be metabolized into its own separate amino acids, which will be released into circulation. So this is just a look at the life cycle of a red blood cell, how the different parts are kind of broken up and where they go for either destruction or elimination, whether that's in your liver, the spleen, or out the body as feces. Some erythrocytes disorders, they're classified as either anemia or polycythemia. So anemia is where blood has abnormally low oxygen carrying capacity, that is too low to support normal metabolism. It's a sign of problem rather than a disease itself. So we don't really characterize it as a disease, but more just as a problem. Fatigue, pallor, gray, pale skin, dyspnea, and chills. 3 groups are based on the cause: blood loss, not enough red blood cells being produced, or too many red blood cells being destroyed. So that's anemia. Blood loss can be from hemorrhage, so we can call hemorrhagic anemia as rapid blood loss. All you do is treat it with more blood. Chronic blood loss anemia is slight but persistent blood loss, usually due to hemorrhoids or a bleeding ulcer. So you're kind of bleeding intermittently, not realizing it, but it's a chronic issue. And the primary problem must be treated in order to stop the blood loss. So stopping the ulcer or the hemorrhoid first. Iron deficiency anemia has to do with not enough red blood cells being produced. It can be caused by some sort of hemorrhaging, but also by low iron intake or impaired absorption of iron from the gut. Red blood cells produced are called microcytes, where they're small, pale in color. They cannot synthesize hemoglobin because there's a lack of iron. So the treatment for this in iron deficiency and anemia is just to take iron supplements. Pernicious anemia is an autoimmune disease. So whenever you hear the word an autoimmune disease, that means the body attacks itself. So pernicious anemia has nothing to do with a supplement or losing red blood cells by some sort of any other, hemorrhaging, but it's an autoimmune disease that destroys the stomach mucosa, so the lining of your stomach, that produces an intrinsic factor. An intrinsic factor is needed to absorb B12 from your diet, and B12 is needed to help your red blood cells divide. So without B12, because it can't get absorbed through the stomach mucosa, because this pernicious anemia destroys that mucosa layer, your red blood cells will enlarge but cannot divide, resulting in large macrocytes. And the treatment is just to inject B12, so get B12 injections or a nasal gel. It can also be caused by low dietary intake of B12, which can be a problem for vegetarians. So if you are a vegetarian or follow some sort of vegetarian diet, maybe think of supplementing with a B12 supplement. Renal anemia is caused by lack of erythropoietin. So this is called renal anemia because your kidneys, renal, are what produce that erythropoietin hormone. This is often the case if there's some other sort of renal disease going on. So your kidneys cannot produce enough of that erythropoietin, which will stimulate red blood cell production. So you just get treated with a synthetic hormone, erythropoietin. Aplastic anemia is the destruction or inhibition of red bone marrow, and it can be caused by drugs, chemicals, radiation, or viruses. All formed element cell lines will be affected with aplastic anemia, and it will result in anemia as well as clotting and immunity defects because your platelet count will be affected as well as your white blood cell count. And the treatment is usually just short-term transfusions and long-term with transplanted stem cells. Premature lysis of red blood cells can affect if too many red blood cells are being destroyed. So we call this hemolytic anemia. That lytic means lysis or splitting apart. It can be caused by an incompatible transfusion or an infection or a hemoglobin abnormality. And this is usually genetic. So if your hemoglobin is abnormal, that's usually a trait that's passed down. And this is where we hear of sickle cell anemia. I'm not as familiar with thalassemias, but sickle cell anemia. Maybe not each kind of anemia, Aaron, but some of them. That doesn't help you guys at all. Let me just go back. Which ones do I want you guys to know? You should know iron deficiency anemia, what that has to do with, just lack of iron. I want you to know that pernicious anemia is an autoimmune disease. So one that where the body affects it, I want you to know basically maybe just the 3 groups of anemia, either they're losing in blood, not enough red blood cells, or too many red blood cells destroyed. So no, not everyone. Iron deficiency and pernicious anemia. I like to focus on those because those are the ones that people probably have heard of. Okay, I'll skip over this one because they probably don't want you to know that. Sickle cell anemia, I do want you to know this one because it's a genetic disorder. And we'll talk about it. If we might not get to genetics too much, but hemoglobin S is just mutated hemoglobin that attacks your hemoglobin cells. So sickle cell anemia, I do want you to know it's a genetic disorder that affects the kind of structure of your hemoglobin and will lead to death. So sickle cell anemia kills 1 million each year. A benefit, people with sickle cell do not contract malaria for some reason, but sickle cell anemia just affects your hemoglobin. All right, you can see kind of the difference in the red blood cells in sickle cell erythrocytes. So the erythrocytes basically look sick underneath a microscope. Oh, Charisse, really? Oh, I'm so sorry to hear that. So yeah, so does it run in your family, Charisse?
Yes, my oldest brother, he was 6 years older than me.
Okay.
I'm the baby of 3. So he had a full-blown sickle cell. And then me and my other brother, we just have the trait. And when he was born, of course, it was a lot of problems. And he was given a certain age to my parents that he was supposed to live to. So actually he was 3 days shy of what they said, which would have been 30.
So did he pass away, Charisse?
Yes.
Oh,
yes.
How long ago was that?
Ooh, it's been
a while then.
It's been 13 years now.
Wow. Oh man, but he made it. Oh wow. So did he?
Yeah.
Was he treated with transfusions?
It was because in the early 80s, he was born in 1965.
Okay.
So in that time, AIDS and stuff like that. So yeah, he had a couple of transfusions, and then when AIDS and stuff came out, it was really scary for us. But he was tested, and luckily, we was okay from that. But he actually had a sickle cell. So he went into the hospital because he was having a sickle cell episode, and then he developed pneumonia. So it just down spiraled from there.
Okay. Wow, Charisse, thank you for sharing that. So you have the trait.
Yeah, so I could never get a spouse. If my husband would have had the trait, we couldn't have been. Well, it was advised to us it's not good to have kids because then they would have had full blown sickle cell. So I have the trait and my husband didn't. So
Okay.
our kids have the trait, though.
Okay.
They do have the trait.
Wow, Charisse, man, thank you so much for sharing. I can't imagine having to go through that and then having that effect, asking your husband you want to know if he has the trait, and then
because if he had that would have, if that was the person I was going to marry and wanted to have kids, it would have changed my whole direction. I wouldn't have done it.
Yeah,
because I seen my brother go through it.
Yeah, you wouldn't want Oh, Charisse, wow, thank you so much for sharing that.
Oh, you're welcome.
All right, so anemia is kind of low red blood cell count, and polycythemia is the opposite. So poly means many, so poly is an abnormal excess red blood cells. This can increase your blood viscosity, causing sluggish blood flow. I won't really have you guys know the differences of polycythemia or secondary, but just know that polycythemia just means an abnormal excess of red blood cells. Blood doping, athletes can remove, store, or reinfuse red blood cells before an event to increase their O2 levels for stamina. So that's the idea of blood doping. If anyone has any more, well, you probably don't want to share personal experiences about that, but I don't know a lot about blood doping or what athletes do, but it's kind of crazy what people will do to win. Isn't that funny? Like just to win as we're talking about the election night, which is a bad pun joke. All right, let me see. I just want to get through I want to get through about halfway through, and then we'll finish this on Thursday. Because blood is extremely detailed and really important. The leukocytes, so there will be 5 types of leukocytes, and I do want you guys to know the 5 types of leukocytes. These are the white blood cells. These are the formed elements. That's a complete cell with the nucleus and organelles. They make up less than 1% of your total blood volume, and they are functioning in defense against disease. They can leave capillaries via what we call diapedesis, where they just squeeze through the capillary wall to enter into the tissue, and they move through your tissues in the body through this amoeboid motion and what we call positive chemotaxis. Leukocytes, so your white blood cell count is over 11,000 per microliter. It will always increase in a normal response to an infection. So if you get a blood sample and they see your white blood cell count is high, that means your body is fighting off some sort of infection. We group the leukocytes in either two granulocytes or agranulocytes, and that's just if they contain these visible cytoplasmic granules. So there's 3 types of granulocytes: the neutrophils, eosinophils, and basophils, and then the lymphocytes and monocytes are the agranulocytes. Here's a mnemonic that I've never heard of before, but hopefully this will help you, because I want you to know the 5 different types of white blood cells. Never let monkeys eat bananas. And that takes you through kind of remembering the 5 different types of leukocytes. Okay, so here we go. In general, the most abundant white blood cell is the neutrophil, and then the lymphocyte comes in after that. But the neutrophils are the most abundant white blood cell that you have in the body. And these are what they look like. So the neutrophils have the multi-lobed nucleus. Eosinophil is a bilobed nucleus, and they usually stain these certain colors because in making these microscopic slides, generally they use the same color of stain when they stain the blood. So that's why when they say red and blue cytoplasmic granules, that's due to the staining. A basophil has a bilobed nucleus, and then the lymphocyte and monocyte, the lymphocytes will be smaller, kind of similar to the size of a red blood cell, and the monocytes are the largest, and they have a kidney-shaped nucleus. So these are all that you can see underneath a microscope. Again, I haven't made your exam yet, but I want you to know, neutrophils are the most abundant, they're multi-lobed, and then lymphocytes and monocytes are the agranulocytes. So just knowing kind of these basic characteristics of the 5 leukocytes. So granulocytes, there's 3 types of them listed here. They will be larger and shorter lived than a red blood cell. They usually have a lobed or multi-lobed nucleus. So just kind of different lobes pieced together. They are all phagocytic to some degree, meaning they'll engulf their material to degrade it with their lysosomes. Neutrophils are the most numerous. They're about twice the size of a red blood cell. Their cell can have their nucleus in their cell can have anywhere from 3 to 6 lobes to it. So neutrophils, most numerous, multi-lobed. They're very phagocytic. They're referred to as bacteria slayers. And that's all I want you to know. So they're phagocytic cells, multi-lobed. They're the most numerous. So here are the neutrophils. There's 2 shown there, and you can know they're neutrophils because they have multi-lobes to them. Maybe I would even have a matching picture question for this one. Eosinophils are much less numerous. They usually have 2 lobes to their nucleus. They play a role in allergies and asthma. So this is what an eosinophil looks like. They release their enzymes on large parasitic worms, if you're ever fortunate enough to get one of those. The basophil is the rarest red blood cell. They're functionally similar to mast cells because they'll release what we call histamine. And histamine is just an inflammatory chemical that will attract other white blood cells to an area of infection or inflammation. So there's a basophil. Then we have our agranulocytes, the lymphocytes and monocytes. Both will have a spherical or circular or kidney-shaped nucleus. The lymphocytes are your second most numerous. There's 2 different types of lymphocytes, the T lymphocytes and the B lymphocytes. And T and B lymphocytes are all both very important for the immune defense. They're crucial to immunity. So your lymphocytes are incredibly important T lymphocytes will act against virus-infected cells and tumor cells, and B lymphocytes are what will produce antibodies. So here's a lymphocyte. And then your monocytes are the largest. They will differentiate into macrophages, which just means they're actively phagocytic. So they will just engulf or eat up their virus or an infection that they need to kind of get rid of. And there is a monocyte, excuse me. Okay, so those are the 5 different types of leukocytes. Leukopoiesis is the production of white blood cells. So erythropoiesis was the production of red blood cells. Leukopoiesis is the production of white blood cells. It's stimulated by 2 different types of chemical messengers, which are shown here, interleukins and colony stimulating factors. This really gets into some sort of like more on the immune or lymphatic system, so I won't go into this too much here. But leukopoiesis just means the production of white blood cells. Here is how the granulocytes are produced. I'm just going to skip over this. You guys don't have to worry about that. And then how agranulocytes are produced as well. Okay. And this whole like leukocyte formation, it just goes back to showing how every blood cell, whether it will go into a red blood cell, or this just shows your white blood cells, but they all stem from one stem cell, and then they'll differentiate into the cell that they end up being. So that's just the basic leukocyte formation. Some homeostatic imbalances, and then we'll probably end here. Let me just double check. This will be a good place after we look at some leukocyte disorders, and then we'll stop here and we'll start with platelets on Thursday. So many hematopoietic hormones are used clinically to stimulate bone marrow. So such as erythropoietin or colony stimulating factors are what will start this blood cell kind of formation of the white blood cells. These will help stimulate bone marrow of cancer patients receiving chemotherapy or stem cell transplants. So these hormones are used clinically for that. They're also used to increase protective immune responses of AIDS patients. We can have some leukocyte disorders, pretty much an overproduction or an underproduction. An overproduction of abnormal white blood cells are leukemias and infectious mononucleosis. And an abnormally low white blood cell count is called leukopenia. Leukopenia can be induced by certain drugs, especially anti-cancer drugs, because when you give someone an anti-cancer drug to kill off the cancer cells, you're also killing off other white blood cells. So that could lead to leukopenia. Leukemia is a cancerous condition involving an overproduction of an abnormal white blood cell. Usually there's a clone of a single abnormal cell that continues to remake itself, so an abnormal cloning. It's named according to the abnormal white blood cell clone involved. So myeloid leukemia involves all the descendants from the myeloblast, and lymphocytic leukemia involves all the lymphocytes. Leukemias can be acute or chronic. Acute usually affects children, quickly advancing, and chronic, slower advancing leukemia involves proliferation at later stages, and that's more prevalent in older people. Without treatment, all leukemias will be fatal because you'll have all of these immature, abnormal, non-functional white blood cells flooding your bloodstream. Cancerous cells will fill the red bone marrow, crowding out the other cell lines. So this will lead to anemia and bleeding. Death is usually from internal hemorrhage or just an overwhelming infection. Treatments can be irradiation, anti-leukemic drugs, or stem cell transplants. Infectious mononucleosis, so this is the kissing disease usually seen in young adults, not necessarily because they're kissing, but it spreads more quickly among adults. It's called by the Epstein-Barr virus. Symptoms of it are tired, achy, chronic sore throat, and it usually runs its course with rest after 4 to 6 weeks. So it's incredibly contagious though. And then I think we'll stop there with platelets.